Dr. George Church is professor of genetics at Harvard Medical School and director of personalgenomes.org, which provides the world's only open access information on human genomic, environmental, and trait data. His 1984 Harvard PhD include the first methods for direct genome sequencing, molecular multiplexing, and barcoding, and this led to the first genome sequence of the pathogen Helicobacter pylori in 1994, which of course is a major cause of morbidity in the GI world. His innovations have contributed to nearly all next-generation genome sequencing methods in companies. This, plus chip-based DNA synthesis and stem cell engineering, resulted in founding additional application-based companies spanning fields of medical diagnostics and synthetic biology and therapeutics. He is the director of the NIH Center for Excellence in Genomic Science. His honors include election to the National Academy of Sciences, the National Academy of Engineering, and the Franklin Bauer Laureate for Achievement in Science. He has co-authored over 370 papers, 60 patents, and one book. Beyond that, I would also refer you to a fascinating and very inspiring profile of Dr. Church that just appeared in the October issue of The Scientist magazine. This actually includes a link to his appearance on The Late Show with Stephen Colbert, where he was asked about resurrecting the woolly mammoth. So he can tell us about that, perhaps. In this article, Dr. Church also recounts his first year as a PhD student in 1975 in microbiology at Duke in the laboratory of Sung-Ho Kim. 12 months and 5 papers later, he decided to switch to a degree in biochemistry, but ended up leaving Duke in part because he found the coursework very redundant from the work he had already taken ahead of time as an undergraduate. Don't get any ideas, students. On the other hand, fortunately, Duke's loss was Harvard and our gain, as he entered their graduate program in 1977, and I think it's fair to say the rest is history. So it's a great privilege to introduce Dr. George Church here.
Yeah, thank you very much. It's great to be back here. This is my conflict of interest slide. And in particular, I tried to pick 5 of them that might be particularly relevant today, but this omits a few of them, and so you can go to the website to find it. I'm trying not to rush this, but that particular point. I think I'd like to give you insight into where I think innovation, since this meeting is about innovation, might be going in our field. And we have to look at a little bit at how hard it is to predict. So these are showing major deviations from a Moore's law curve. In fact, both reading and writing genomics has greatly exceeded expectations of even the very steep Moore's law exponential. And here you can see that we can both read and write human genome as sort of in 200 base pair chunks, so not a complete one, for on the order of thousands of dollars. And you ask what happened in 2003, 2004 that caused that huge change in exponential. And I would argue that these 2 papers that basically took the lessons from semiconductor industry, and we started reading and writing DNA by miniaturizing them on chips rather than moving them through capillaries or fluidics and that sort of thing. And it also illustrates this intimate dance between synthesis and sequencing to the extent that we even call the methods that one of these 2 papers sequencing by synthesis. And furthermore, a lot of the synthesis we do is key. It has a key underpinning. It's hard to do editing, for example, without knowing the genome that you're editing. Now, what do you do with a whole human genome's equivalent of synthetic DNA? I mean, you might say, well, what's the point of that? I mean, we know the point of sequencing whole human genomes, and we all wish that all of us were sequenced. But these are examples of applications of whole chip libraries. Whole genome level libraries from chips. And these include short hairpin RNAs used for interference, targeted sequencing by selecting DNA, Barcodes can be used for almost all of these things, where you put in a very clear digital barcode in the DNA. You can have peptide epitope libraries for studying the immune system and other functionality, guide RNAs for CRISPR and cis regulatory elements, and so on. Now, I'm not going to review CRISPR here. This is about the future and innovation. CRISPR is so old now, 4 years old, that it's no longer innovative. And maybe it never was, because it was just another nuclease. But it is important to be clear on why it was innovative, considered innovative. And a lot of people think it's just because you have to make a 20-mer. You just have to design a 20-mer that's unique in the genome, and then that will cause a cut. And that's ease of use or cost. I don't think that's the case, because about the same months that we were introducing CRISPR as a technology, this paper came out from Junsu Kim's lab in Korea that showed that the previous nuclease, the Talon class of nucleases, which was completely synthetic, Talons don't exist in nature. They're combining two bacterial proteins. But anyway, this lab just kind of calmly just spun off a paper. There were 18,000 different nuclease and cutting sites were described. So clearly that was fairly easy to do even back then. I would argue instead it's part of another one of these exponential curves that has to do homologous recombination efficiency, sometimes called editing. And this dates back to Oliver Smithies and Mario Capecci, who started this in 1985 and eventually got the Nobel Prize for efficiencies around 0.1%. And then it bumped up a little bit to maybe 0.7% for Talons, and then 8% for CRISPR. So these are fairly small increments. And then there's one I'll show you in a moment from Luhan Yang's work, where it went up to not 62%, but 62 instances in the same genome. We'll see that, and it was done in an odd setting. And so what enabled that innovation? What enabled that to work? What enabled that bump in maybe 20-fold from Talons to CRISPR in homologous recombination? And that's a particular kind of editing. I would argue that it was this guide RNA. This was the key thing that wasn't obvious. We had 3 guide RNAs at about the same time. You can think of it like Goldilocks. The one at the top was missing its 3' end and missing a motif that the CRISPR protein had to recognize. The one at the bottom had that motif down, but was missing the a fusion between the two halves, which isn't necessary in bacteria, but apparently is extraordinarily helpful in mammalian cells. And then the one in between was just right. It had both the fusion and the 3' motif. And this shows the consequences of that. In the unfused case, you had sort of homologous recombination rates, not too different from the Talons, about 0.7, or 0.37 for the Talon here done side by side, going up to 8% with the single guide RNA. So that's a factor of about 22-fold between Talons and guide RNAs, and on average it was closer to maybe 4-fold improvement. Now, what was that case of not sort of 8%, but essentially 6,200%, 62 genes simultaneously? And this was motivated by a long-standing problem, long-standing innovation. 20 years ago, it was innovative to consider the idea of transplanting organs from pigs into humans. It was clear that you had to do some humanization, not just in terms of immune function, but the complement cascade and the clotting cascade. And so there was a hope that you could do this with a single gene or maybe 2 genes, and each year was another gene. But then suddenly people realized that actually, in addition to all that humanization, there was this problem of endogenous retroviruses being produced by the organs in the immune-compromised patient, infecting human cells, and incubated in this immune-compromised patient, it could go badly, not just for the patient, but the evolution of a new zoonotic disease like swine flu. So anyway, that kind of put a kibosh on a $1 billion investment 15 years ago. And so when we re-entered with our new hammer, everything looked like a nail, which was CRISPR, we said, let's tackle that first. And we found that there were 62 endogenous retroviruses, or PERVs. It's not my acronym. 62 we had to hit at once. And that was pretty challenging, even for the arrogance of the CRISPR field. But we tried it, and it was not exactly hard work. You put the cells in the incubator. 14 days later, you screened them and found that they had knocked out all 62 endogenous retroviruses. So that still is kind of the record for this. Luhan Yang led this effort, and now she's just recently set up shop at a little company called Epigenetic to do all of the humanization and retroviruses. Now, this technology you can go more. You can go beyond 62. In fact, we were so surprised how easy that was that we're now tackling all sorts of, essentially every major class of repetitive element, the signs, the lines, and the centromeres. Furthermore, you can get it so it can go at close to 100% efficiency in the wild. So not just in one cell or one organism, but you can drive it through the wild. I'm not going to go through the mechanistic details because this is a short talk, but I'm happy to. And it can be applied to all these different societal needs, most notably the controlling vector-borne disease for things like malaria and Lyme disease, where you can either spread through the population, a time bomb that will cause a crash in the population of the vector, or send through it a resistance gene, like multiple antibodies that cause it to be resistant to the disease that it carries. So we did an unusual thing in this case where we wrote a paper about the theoretical promise and risk of this new technology, and various ways of preventing it escaping from the lab or allowing reversal once it's in use. And most recently, we published a paper called Daisy Drives, where we can constrain this to a geographical location, which was totally not obvious when we started this. Here's another interesting application of CRISPR that's kind of perverse in a certain sense. You can see that kind of CRISPR 101 is on the left here. You transcribe the guide RNA, which has kind of a blue and a red part, but it's all one guide RNA. It's transcribed and it binds to the protein, the CRISPR-Cas9 protein, which then cuts a target down below. So you transcribe the guide RNA as one piece, and it cuts. And then once it cuts, it damages the target, and the target is no longer a target. Simple, okay? But what if the thing that you're transcribing is itself a target? That is to say, you include that little golden region, which is the so-called PAM, which is normally encoded by the protein and normally not in the guide RNA. But if you put it in the guide RNA, when it cuts itself. It now transcribes a new guide RNA, which cuts itself again. And you might say, well, okay, this is going to generate a crazy number of different guide RNAs and different genomic sequences and encode them. What good is that? And what we developed it for was for so-called homing barcodes. I mentioned barcodes earlier. This is something where, unlike the barcodes that you make by synthesizing DNA on a chip, where the computer has designed them to be orthogonal and clean, digital identifiers, this is something where the cell is essentially generating them as the embryo develops from a single fertilized egg, single cell, into hundreds or trillions of cells. That means that every cell division you generate diversity, and you can track essentially the pedigree or the phylogenetic tree of that developmental lineage. And there's very few really developmental lineages for any animal outside a few nematodes. And so this would be a tremendous resource. And Reza Kalhar, postdoc in the lab, developed this along with Prashant Mali, who was one of the co-inventors of CRISPR, both for human cells and for mouse embryos. And so this kind of ticks off two different things we have here. That ID, that barcode ID that's generated on the fly, is one of a class of different strategies where we would like to be able to record biological information in a compact form into DNA. And it allows us to get specific IDs during the developmental lineage, as I mentioned, but also in the connectome. If each cell has its own unique barcode, whether or not it's developmental lineage derived, you can say at a particular synapse which is the postsynaptic and presynaptic, and which cell is connected to which, even if their synapses are centimeters away from the cell body. And this is part of kind of an overarching set of goals that we want to integrate in this so-called brain project that I had the pleasure of helping with in the very early days. And I loved the Obama's team's acronym because it ends in the words innovative neurotechnologies. Again, I think it's tremendous to have a project of that nature. Now, if we're going to work on human data and cells, we need to be able to share it. And there's one fairly unique cohort, which is international in all these different cities around the world, where we have IRB approval, or the equivalent different acronyms in different countries, to be able to share this vast amount of data per person. Because the more data you have on a person, the more it's like what we think precision medicine will be, but it's also more identifiable. And so these people know what they're getting into. And sharing human cells in particular, it's hard to encrypt a human cell. So this has allowed the first example in the ENCODE project that Manolis mentioned earlier. This is the first set where you have 3 different cell types, vastly different epigenetic states, all from the same individual. So fibroblasts, stem cells, and neurons. And those were studied by all different kinds of chromatin and transcription analysis. And the National Institute of Standards and Technology, FDA, and Personal Genome Project, from which this cohort came, led to standards for genomics called Genome in a Bottle. So if anybody wants to develop a new diagnostic or instrument and so forth, they can get, there's tens of thousands of these identical bottles, and they're from many ancestrally diverse trios. There are many different ways that we can produce organs. We want to produce organs not only for testing new therapies, but also for transplantation in its own right. I gave you an example of that already, where you can humanize pigs for transplantation. And then there's various other ways, and the people in the group that are working on getting these organs. I'll give you an example where, in addition to animal models, we can have human, increasingly faithful and representative human organ-like organoids. Here's one for cardiac tissue where, with Bill Poo, who did the clinical genetics, had a potential hypothetical cause for this cardiomyopathy, which was a missing G on the X chromosome in the TAS gene. And this was, as we saw earlier, there are often many candidates alleles. There are 3 million differences between you and me, and between me and this child. But we wanted to make a clean experiment to ask, and we'd like to have a pipeline to ask in general, these variants of unknown significance, which one's actually causative rather than merely correlated. And to do this, you can use the CRISPR or other editing methods with one of these properly consented cell lines from Personal Genome Project #1. So that Cas9, with a repair oligo, you get this homologous recombination. If you leave the oligo out, you get a mess. I don't really call it editing, call it genome vandalism or non-homologous end joining. And if you leave out both the Cas9 and the oligo, then you get a control. And then you can So that's genetic alteration, and then you can do epigenetic alteration after you've established a clonal cell line where you know that just that one base pair has changed, nothing else in the genome. So no matter what the off-targets are, you can use this trick of using a clonal cell line. And so here's 3 different clonal cell lines that have been turned into these pretty remarkable cardiac-like tissues that expand and contract in a pulsatile manner. And you can see this beautiful repeating sarcomere organization in the normal control, but then one base pair deletion, 1 out of 6 billion base pairs changed here, abnormal morphology and physiology. And we're working with groups, and we're happy to work with anyone to establish pipelines for doing this sort of thing on a large scale, maybe millions of variants of unknown significance. We already have pretty good diagnostics by whole genome sequencing, but this would take it up another notch where you can get at causalities for all these different motifs. Now, when we talk about genome editing, there's a lot of talk and a lot of action, too, on solving the problem of off-target cleavage. This little cartoon here shows one of the many strategies of requiring 2 nicks near one another in space, so it's kind of like a coincidence checking mechanism. So that's paired nickases. There are ways of weakening the binding to one or the other strands here. And each of these comes out from my lab and a colleague's labs. We come out, we'll say it has close to nothing detectable off-target. Now that sounds very impressive, but what it means is you've computationally designed them so that there's nothing in the genome that's off by 2. It's off by 3 or more. And that means it's really not that challenging a task. And there are many cases where you actually care to be able to distinguish off by 1. That is to say, a single nucleotide polymorphism that might occur between 2 alleles that are inherited or 2 alleles that occurred during cancer. And so the first SNP-specific method is not in a high-profile journal yet, but if you're curious, take a look at this bioRxiv paper. That's off-target. But what's often not recognized is there's a lot of on-target problems with CRISPR. And so I call this one of the examples of beyond CRISPR, where we need to look beyond, and we are moving beyond, as you'll see. And the real problem is you get this stuff that I referred to as non-homologous end joining or genome vandalism, where there's a race between when you make a double-strand break and you bring in your patch editing oligo, the cell might get there first, and it makes insertions and deletions. And so there are various solutions. Like I like the deaminase solution, where you actually make a single atom change from a nitrogen to an oxygen without making any double-strand breaks. Our lab worked on this. David Liu's lab did a nice Nature paper, and there are other groups coming out with it now. And then these 2 methods, which are very popular in the prokaryotic genome engineering world, where you'll have a site-specific recombinase. This is almost the ultimate one, where you don't do any cutting at all until the donor and recipient molecules, DNA molecules, are touching one another. So you don't leave a double-strand break free in the nucleoplasm for the cell to do what it wants. And then there's another one that psychs out the replication fork. And I want you to hold those 2 mechanisms in mind, because in the next slide, we have a project entirely based on those 2. No CRISPR used for recombination at all. Instead, we can, beyond CRISPR, this is the largest genome engineering project I know of in the world right now, where we did 4 million base pairs of we got excited about 62 base pairs a little while ago. This is 4 million. And it's done by a combination of those 2 recombinases. And the goal was, many goals, but in particular, multivirus resistance by changing the genetic code. Now, we can disable the CRISPR nuclease in order to turn it into an epigenetic machine. It's something that can inactivate or activate genes or otherwise modify chromatin. And here's the latest of the various ways of doing this, where the dCas9 or dead or defective Cas9, no longer nuclease, still binds the guide RNA, binds the genome, but it has 3 different activation domains, 3 different ones in a particular optimized in size and order so that they have the maximum impact. And you can see that when we just had one activation domain, and this is a pretty highly optimal activation domain to start out with, and a fairly, you could get excited about a 63-fold effect, but wow, you can get a 20,000-fold increase by putting all 3 together rather than just one at a time. And this is for a very large, this is the largest protein coding region in the human genome. Just forget the introns, just the coding region itself is 100 kilobases. Here's another gene, neurogenin 2, where we wanted to get activation, changing the stem cells here in blue, kind of all piled up into these much more elongated neurons in red. We wanted to get that to be as efficient as possible. You can see here we're getting it up into the tens of percents, but still, even though we're getting these huge induction, that's not enough to get full conversion from one to another. So here's another beyond CRISPR innovation where we can, at least in many, many cases, we can, for reprogramming cells epigenetically, we can go further and faster using a transcription factor library. To our surprise, there was no transcription factor library for humans or any other higher eukaryote. When we looked at this, and Alex, a graduate student, slowly put together a full set for all the human transcription factors and is making this available to a lot of people pre-publication now, and it will be distributed soon from adgene.org, as is almost everything I've described in this talk, freely available through this nonprofit essentially at cost, $50 or something like that. And this is an example of an experiment where we screen the entire transcription factor library looking for both pluripotency, things that de-differentiate or keep in a non-differentiated state, all the way to differentiation into a whole variety of things. And remember that last slide where there was very poor conversion from stem cells to neurons? Here we can get 98% conversion in 4 days with a variety of different transcription factors we found, and either into very specific bipolar neurons that have just 2 processes or 3 or more. And so we're building brains and various parts of them in this way. Now, in order to validate these artificial tissues, we need information that doesn't exist, and we need a general method. So I mentioned ENCODE. ENCODE and GTEx, if you know the jargon, they typically involve mixed tissues. They involve averaging over many different cells. We need single cell and, in fact, subcellular resolution so that we really know that the cell we created is not just, because if you look at a chunk of tissue, you've got all kinds of different cells in there. In addition to the tissue-specific cells, you've got endothelial cells, blood cells, and so on. So anyway, so we want to have all these ohms integrated and in situ. You want them essentially as an extension of your microscope. You want every pixel or voxel in your microscope to have a name, the naming the molecule is that point. Here's an example of 2 cells where every of the 4 colored dots is a single step in a sequence, and each one is a clean color, which indicates that they're clonal and they're producing clean sequences, you'll see in a moment. So this was something that was in the works since 1999, but finally Jay Lee, a postdoc who's now at Cold Spring Harbor, and Evan Doherty, who was a graduate student, pulled this off, and it's and you can see here in a single cell, each of these dots is changing color because we're going through cycles of sequencing. It's a little sped up from real sequencing, and then you can replot it where each point in the cell is now a tag. It's a sequence tag, and here we mean sequencing quite broadly. This is now a company that offers either the instruments and the reagents, the whole system, or a service. And you can see here the clean separation of G's, A's, T's, and C's, these 4 different colors. We've used it as a discovery tool. Here is the first paper in Science. You can do the usual systems biology. You can discover point mutations SNPs, RNA editing, alternative splicing. Here's an example of something that's missing in one media and found in another in this wound healing experiment on human cells. You can do not only RNA, but DNA at super resolution. This is work from the Wu lab called oligopaints. You can do proteins as well here. And this illustrates two things from Ed Boyden's group with which we've been collaborating for years. You can not only get expansion microscopy, where you literally can expand up to 8,000-fold in volume, so 1,000 reads can turn into maybe 8 million, but you can also resolve proteins that are hard to resolve. Here are postsynaptic and presynaptic proteins that have been labeled with nucleic acids so that we can turn all the DNA, RNA, and protein into a nucleic acid assay. You can also, we're just beginning to do this. This is a little speculative, kind of, we have made ligand-specific proteins from scratch. These are not naturally occurring for two different plant and animal steroids. So this is progesterone-specific and not for all these other closely related chemicals. And then you can make a protein dependent upon that for its stability. So now you've turned a ligand problem into a protein problem, which we've already turned into a nucleic acid problem. They can all be detected by variations on fluorescence in situ sequencing and hybridization chain reaction expansion fish microscopy. So I just want to make sure we have time for questions. This is, in addition to all the acknowledgments I made along the way, these are 4 of our major grants. This is a particularly fun Center of Excellence in Genomic Science from the NHGRI on genomically engineered organs with Ed Boyden, Jennifer Lewis, and Ting Wu. And then we've got a couple of brain initiative projects and 2 transformative awards. So thank you very much.